I'll save some people babysitters too and stuff. So we are doing our chapter 24, the urinary system, and you guys are doing kidney dissections in the lab. And so almost everything that I go over, kidney anatomy, should be something that you should definitely make sure that is part of your items that you actually list during your eval. I won't go into too much blood vessels, but I will go somewhat into those. It's important that you realize our urinary system is very different from the cat's urinary system. When you guys go into the abdominal pelvic cavity of the cat, that bladder is right there. It's in the inferior aspect, but it's right out in the open. If you're actually undergoing a surgical procedure like a laparoscopy where they stick the tube inside you, blow you up with carbon dioxide, and look around to take a look at your organs, you will not see a bladder. There is no bladder that you can see inside the human being unless it's distended and full of urine. And even then you can't see it because it is retroperitoneal. And that's the entire urinary system is retroperitoneal. So that means outside or superficial to the abdominal pelvic cavity when we talk about things being retroperitoneal. So keep that in mind as you go through the human anatomy here. That's interesting. I don't know where that came from. Our kidney anatomy here, it says, again, retroperitoneal, reinforcing that everything is retroperitoneal. But our kidneys will be on the posterior wall of the abdomen and will be ranging from around the 12th thoracic vertebrae to the 3rd lumbar vertebral area. And I want us to take a look at this cross-section here of a person. And it's easy to figure out this is the anterior side, this is the posterior side because we see our vertebrae right here. But it might not be so easy for you to figure out which is the right side and which is the left side unless you have an organ that tells you this is definitely on the right side or definitely on the left side. Do you guys have an organ that you can easily identify? Yeah. Yeah, IVC, but what I like is I like the big stuff. I like the big stuff, exactly, the liver. And the liver is predominantly a right-sided organ. We do have a left lobe to our liver, but it's nowhere near as big. So here is the liver right here. And so right off the bat, we know that this side is the right side, and then this side is the left side. So whenever you are, because you will be, it's amazing that you will be looking more and more at cross-sectional anatomy as you get into your careers, because we do a lot of CAT scans and a lot of MRIs, not as many x-rays as we used to. So CAT scans and MRIs. The other thing that helps me with right and left is this guy back here, and that's the spleen. The spleen is always a left-sided organ behind the stomach. And so indeed, our stomach is right here anteriorly. You know that this is the pyloric sphincter right in that area, correct, from this view? And so therefore, you know that this is the beginning of the small intestine or the duodenum or duodenum. Either is just fine. It's nice to realize our aorta is fairly midline for us, but our IVC, our inferior vena cava, has a tendency to be to the right. So that's something to also realize too, because it's the same way for the cats. The IVC is to the right of the abdominal aorta. We know that this is the large intestine, and because we're by the spleen, we know that this is going to be our descending colon, or maybe even a portion of that area that was close to what's called the splenic flexure. So as we take a look at this. The pancreas right here is indeed right behind the stomach. And then here are our kidneys, because we've identified most things here. This would be our transverse colon based upon its location. So we have a kidney here and we have a kidney here. And one of them looks larger because this slice is going more through what I call the majority of the body of the kidney, and one is at more of a superior aspect of the kidney or inferior aspect. And so over here on the right side with this right kidney, it looks smaller than the left, but it's not really smaller. It has been displaced inferiorly, so this is a more superior portion of that kidney because the liver is such a big organ. Remember, it's our biggest internal organ of our abdominal pelvic cavity. So it's displacing the right kidney a little inferiorly, so the kidney that one will be closer right to the 3rd lumbar vertebrae. And then our left kidney is therefore a little more superiorly in its location and would be the one that's starting around the 12th thoracic vertebrae in that area of the abdomen. So that's one of the things to do, and I think it's kind of nice to be able to take a look at some of these things and start to realize the anatomy that you guys have actually been learning and are doing really good with. Let's go to the external anatomy listed here. Hilus is the medial indentation. Hilus is the same as hilum. Hilus is older, and unfortunately I'm older and I use some of the older terminology. But hilus and hilum are the same. It's singular. It's not actually a plural term. So I just want you guys to realize if you see an S or an M, it's the same thing. At the hila aspect, that's medial indentation, so our little beans are facing medially, so to speak. And from that area, we will find our ureter and BVs exiting or entering, depending upon your structure, right? The ureter exits, the vein exits, and the artery enters. The adrenal glands are in the fat at the superior pole or superior end of each kidney. They sit on top of our kidneys and have a tendency to drift a little bit medially. But because of that location, they also have another name called suprarenal. So they're the suprarenal glands. The kidney itself has a fibrous capsule. Its thin, dense connective tissue. Is it regular or irregular in the arrangement of the fibers? Irregular, because we want to make sure that if there is for any reason some sort of expansion of the kidney, that the capsule is not going to split like a banana peel. So indeed, irregular would be the arrangement of our fibers. We also have perirenal and perinephric fat. Nephron is a very important functional and structural unit of the kidney, and so you'll hear nephric a lot as we talk about kidneys, or renal also. So perirenal fat, perinephric fat, it cushions and surrounds each kidney because our kidneys are not locked into place very tightly. They have a lot of what I call wiggle room, and so that fat is there to actually help to protect them and give them some support. We also have stuff called renal fascia. Fascia is the material that surrounds our muscles and stuff like that, kind of keeps them properly bound up. And so we have also some fascia similarly, so it's got some good structure to it, that is designed to keep our kidneys in place. And when we take a look at this picture here, this is what we're talking about. So inside that fascia, there's fat, and outside that fascia, there's fat. So, and that's what you guys found when you were dissecting out your cat's kidneys. They were definitely embedded in this fatty area, and you had to get rid of the fat in order to actually expose the kidneys and start to look at their structure, and which isn't too terribly exciting, actually, an external aspect. But on the kidney itself, the only thing that you guys are going to be able to identify from this list is the fibrous capsule for your pig kidneys in lab. Then as you open up the kidney, you bisect the kidney and splay it open. There's a definite internal structure that you'll be able to identify. And three main regions are actually in this area that is easy to pick out. The cortex, the light-colored outer region, and it's light when you compare it to the medulla or medullary area. Inner region darker in color, and the medulla will have renal pyramids as well as renal columns. Those are both making up the medulla or the medullary area of the kidney. Now, we're going to draw a pyramid, dark cone-shaped masses, and they are upside down. So that's a pyramid. So this little spot right here is our apex or papilla. This area here would be called the base. Even if it's upside down, That broad area is actually referred to as the base. From this pyramid and the papilla, you will actually have the final formation of urine. So urine will be drip, drip, dripping from that little tip right there. And we have to collect it. It has to go into a vessel. And the collection vessel is called a calyx. And so I'm going to draw a calyx here. And we have several of these pyramids. And so let's draw another pyramid. And let's draw another calyx, and these calyxes come together. So these are indeed, the calyxes are part of the collecting structures. What's the first calyx called? That's correct. So it's a minor right here, and then where the two come together, that's a major. So all this area is major. And then the major calyx will empty into the pelvis, which will then empty into the ureter. So as far as collecting structures, there's only 4 collecting structures. But the medullary area is indeed renal pyramids, and you can actually identify various things in the renal pyramids, the base and the apex, for example. Now the renal columns, they separate the pyramids, and you will find blood vessels here. And these blood vessels have a specific name, and they're called interlobar. So you'll have an artery and a vein in those renal columns. Interlobar artery, interlobar vein. And your kidneys are injected with latex. Red for the arterial system, blue for the venous system, and there'll also be yellow latex that's inside the collecting structures itself. The cadaveric kidney over here, easy to still see the basic structures that we have been talking about. Don't forget this area is the hilus of the kidney. Again, this is the cortex, the medullary area. We have a calyx here and a calyx here, pelvis here, and ureter here in this area. And so still we RC, renal columns, again, apex, base. So those things, as you are working on getting yourself ready for your eval, you should be able to start labeling various areas of the kidney quickly so that you can easily get this dissected out and some structures identified. Renal artery and renal vein obviously are going to feed or drain the kidney. It's important that you realize that the renal artery actually starts to branch often before it enters the kidney, and that first branch has a name. All branches of any vessel will have a new name, and those are segmentals. You guys know what arches over the base, what blood vessels name arches over the base of the pyramids? Arcuates, yes. And we also have blood vessels that are going to radiate into the cortex, and they're called cortical radiating blood vessels. We prefer that name because there isn't a name that's been used, interlobular, but cortical radiating blood vessels actually are much more descriptive, don't you think? And interlobular, where's the lobules or little divisions, and you can't actually really appreciate that. So some books will call them interlobular. We're going to call them cortical radiating because that's a good descriptor for us. Again, going back to a collecting structure, renal pelvis, funnel-shaped tube that collects urine. Not much there to that we want to add. I call it a wide spot in the road. Before the ureter. Minor calyces, cup-like structures enclosing the papilla of a pyramid based on our notes. I say see above. I'm going to take this space out. And a major calyx, also see above. Blood and nerve supply. What's fascinating, those two little beans in our body, deliver about 25% of the cardiac output at any given time. So it's amazing. They are getting a large volume of blood because they are very important in filtering the blood and monitoring the homeostasis for your system and making sure the fluids and electrolytes and blood pressure and blood volume are all working fine for you. Because when people have kidney failure, all of that goes out of whack. So the kidneys are very important in taking care of a lot of homeostatic function for you, and that's another reason why they get a large amount of blood volume actually sent to them to make sure that it filters it and maintains blood pressure and proper pH, blood volume, and all those other things. Renal plexus. We've talked about plexes before. That's a network of nerves, and this network is of sympathetic nerves, and they are designed to control arterial diameter. Remember, arterioles are right before capillary beds. And so when you clamp down on all these arterioles, then everything slows down going into the kidney, correct? And so the kidney then gets fed less blood. So once you start clamping down on those little babies, everything slows up going to the kidneys, and then that blood is diverted into other areas. And so this renal plexus is important in controlling blood flow to the kidneys. And what do you think? Do you make more urine or less urine when you clamp down on that blood flow? Less. And that's an important thing. So this arterial diameter clamping on down will indeed cause you to actually produce less urine. Now the nephron, the thing that does all the work in the kidney, not really, but well, actually it does most of the work in the kidney, is called the structural and functional unit of the kidney, and we have 1 million nephrons per kidney. These are microscopic blood processing units. And they have a very important tubular system. That we're going to go over. This picture right here is perfect to review before you do your eval. I think it gives off all of the blood vessels as well as the structures that you should actually be naming for your kidneys, if you have not done it already. This area is lighter in color, so indeed it's the cortical area. This area is darker in color, indeed is the medullary area. We're taking a look at the cortical medullary junction right now. And therefore, this blood vessel is called the arcuate. So those BVs are arcuate blood vessels, and they give rise to the cortical radiating ones. Branching off of that, and then branching off of the cortical radiating artery is an arterial that has a very special name called the afferent arterial. And this takes us back to the nervous system, right? Afferent and efferent pathways. So that means going into a structure, and the structure that it's going into is the glomerulus. The glomerulus is a very sophisticated capillary network that is all bunched up into a ball. If we have an afferent, we know we have an efferent. And indeed, we have an efferent arteriole leaving a capillary bed. Have we seen this before? In any of our capillary beds, have we had an arteriole leaving a capillary bed? No, it's actually supposed to be venules, correct? And that's because in most capillary beds, there's always this nutrient and waste exchange actually occurring. In this system, we don't want to have nutrient and waste exchange. We want to filter your blood. We are going to filter your blood, and then we're going to process it. So we want to filter as much of your blood as possible. And that's why we have an arterial, capillary bed to arterial. And I want you to notice the diameter of the afferent compared to the diameter of the efferent arteriole. And they are indeed different in size for a very specific reason. The afferent is much larger in diameter than the efferent, which means, therefore, the pressure inside the glomerulus is what? Higher or lower than most of our capillary beds? Higher. And that's important because these guys, always trying to come back and haunt me, these guys are indeed under higher pressure, but they're leakier. And that means if you're leakier, that means that you're going to actually, under higher pressure, potentially actually destroy them. And so this is an interesting system that has its backup to actually protect it, and we'll get into that in just a moment. Going from the glomerulus, so this is like, you know, taking your kitchen rag and squeezing fluid out. Well, it's not a good idea to squeeze fluid out into the air, is it? So we have to squeeze fluid out into an area or a space, and the space is the capsule. And so we actually have here glomerular capsule, so this is that space around it. So it is designed to collect what is called filtrate. This is not urine yet, this is called filtrate. So where we first form filtrate is at the glomerulus and the glomerular capsule. When we talk about the glomerulus and the glomerular capsule together, we will also refer to them as renal corpuscle. So that's a body. A renal corpuscle is the glomerulus, which is a capillary network, and the glomerular capsule. So fluid, filtration, and collection unit right there. Then the fluid is going to flow down our nephron. This whole thing is that we're starting with the renal corpuscle as the beginning of our nephron, is going to go down into the PCT, proximal convoluted tubule. So it does indeed, it looks convoluted, all kinds of twists and turns. Then, if you follow this tubular system, it will go down into what's called the nephron loop. And look where the nephron loop goes into. Look where we were actually in the cortex with all this, and now that nephron loop is going to dip into the medullary area. That's a very, very important area because we're going to go over this for how we make our urine concentrated. This area is key to helping us to make concentrated urine. Then we follow this, and we are now into the DCT, distal convoluted tubule. Again, we go into the cortex area, and it goes all over the place. And the end of the nephron will be in this area at now the CD, or collecting duct. So we have a bunch of nephrons emptying into a collecting duct, and we have a bunch of collecting ducts. And as you can tell, where these collecting ducts are located, the collecting ducts are located in our medullary pyramids. And so this is going to go all the way to the tip of the medullary pyramids, and out of the collecting duct and out of the tip of the pyramid will be the final product, which is called urine. And so we have to go through the process of actually making urine. Reviewing the notes real quick, renal corpuscle, the glomerulus plus the glomerular capsule. The glomerulus is a tuft of capillaries at the beginning of the tubule. The capsule surrounds the glomerulus and is continuous with the renal tubule and is specific to collect the first filtrate. So that capsule is where our first filtrate is formed. Then when we look further at the renal tubular system, where it's actually real tubes, we have the PCT, proximal convoluted tubule. You will have cuboidal cells with microvilli. What's microvilli all about? Increases surface area for absorption. So you guys have learned so much stuff that you already know. When you see these things, you already know what's happening in these areas. And I know you take it for granted, but you have really actually learned a lot. So PCT, proximal convoluted tubule, cuboidal cells with a bunch of microvilli, and this is where our main reabsorption of solutes occur. So this is going to be glucose, ions, vitamins, all kinds of things. We'll talk more about that in just a moment. But main area for reabsorption of solutes. Well, when we reabsorb solutes, what always follows solutes? Water. So also main area for reabsorption of water. And what's absolutely fascinating, these little nephrons, 1 million per kidney, actually make 180 liters a day of filtrate. It is crazy. And how much urine do you usually put out? A liter to a liter and a half. 180 liters of filtrate a day. And so that's what's going into that glomerular capsule. And so this tubular system is also is vitally important for reabsorbing what you want to keep. So it's really strange. You put all kinds of stuff into the filtrate, into these nephrons, and then they start cherry picking. Okay, this is what we need, and all this water is going to go back into your system. And so that's why this is a very fascinating process. I can't tell you why this process came about, but I can tell you what it does and that it works really well when it's working. The loop of Henle is also referred to as the nephron loop. So yeah, I have to tell you the old-fashioned name, loop of Henle. And if you're talking to an old-fashioned nephrologist or kidney specialist, he'll talk about loop of Henle's too. So you need to know that nephron loop is the loop of Henle. That's going to be mostly simple squamous epithelium, and we're going to have a descending limb and an ascending limb. It's very important that you realize these are two very distinct areas where different things actually happen. But our loop of Henle, always a portion of it will go into the medullary area. And I'll say pyramid because that's the medullary area. Then we have a distal convoluted tubule, cuboidal cells without microvilli. So we know one of the things that's important is that this isn't the main area for reabsorption of solutes and stuff. And we can now, with this information, we can go and look at the histology drawings, which on mine is way off to the right-hand side of this screen, but yours are not so far apart. Yours are probably in the middle of your page, but that's the way it goes when I put it on this stuff. So this is the glomerulus, right? And then this is the space of the capsule. Same thing here, up here, that's that space. And this is the parietal layer. Whenever we have spaces that have a limiting epithelium, it's the parietal layer, and the parietal layer is very similar to what we have in our abdominal pelvic cavity or our thoracic cavity. What are those cell types called when you take a look at those? Simple squamous epithelium. So that's very, very normal. What we don't yet see, what we'll see in just a moment, there is a visceral layer that's over the glomeruli. And remember, these glomeruli are under very high pressure, and we'll learn 60 millimeters of mercury, which is almost twice what we actually have in our normal capillary beds. And they're leaky and they're fenestrated, and we'll get into that in the notes too. But they have this visceral layer that are called podocytes that are actually there grabbing onto actually the circumference of each capillary, actually shoring them up and reinforcing them. We'll see that in a moment. This area right here is called the juxtaglomerular complex or apparatus. Juxta. We're going to learn that that's going to be our afferent arteriole and a DCT in that area. So you know that the glomerulus has an afferent coming in and an efferent going out. So there's actually, if you did a proper slice, like if you had a proper slice through apple and you got the stem, that's actually what this is showing you. This is showing you a little bit of that stem area of that apple. From these two views, they're both very different stains for very different reasons. I am going to tell you that you can easily pick out the distal convoluted tubule and the proximal convoluted tubules in both of these views. And I'm going to tell you the reason you can do this is because the proximal convoluted tubule have a bunch of microvilli, which means that inside the lumen, it's fuzzy looking, adjacent to the luminal aspect of the cells. It's fuzzy looking. And I'm telling you the distal convoluted tubule doesn't have any microvilli, so it's not fuzzy looking. Now that I've told you that, let me ask you, what is that? Distal convoluted tubule. And it's easy to figure out that's one, and this is one, and this is one. And so all the others are proximal convoluted tubules. They are fuzzy looking on the inside in their luminal stuff. And now that you know that, to help you pick it out, you can also appreciate the cytoplasm is staining differently between the two. So that means there's different things going on when the cytoplasm stains a bit differently. Now let's go down and take a look at the inferior one on my screen. And let's pick out, I'm going to erase some of this stuff. And let's go ahead and ask you, what is that? That's a distal, so therefore this has got to be a distal. This is a distal. And so then it's easy to figure that's a proximal and this is a proximal, because luminally the distal is pretty clean looking compared to the proximal convoluted tubule. And then also, again, we notice that there is a differentiation in how they take up the stain. So that is an easy thing to actually figure out histology-wise. Renal corpuscle, glomerulus plus the glomerular capsule, parietal layer, simple squamous epithelium, visceral layer. They're called podocytes. Podiatrist, foot doctor? So podocytes. And that's because those cells have foot-like processes that are designed to form filtration slits around the capillaries of the glomerulus. And so when you take a look at this cartoon over down here, you see these little podocytes in their nucleus right there, and you see what looks like branching frond kind of leaves, you know, reaching out. And that's what they do. They reach out and they surround that capillary and shore it up. They shore it up, and because they have these little finger-like or foot-like processes, they will interdigitate and create those filtration slits, because these capillaries are fenestrated. Fenestrated capillaries means that they are leakier than the average capillary. So we have a couple of factors here making these capillaries very leaky to create filtrate. We have extra leaky capillaries and we have higher pressure. And so we'll have higher hydrostatic pressure pushing fluid out into the capsule. And so that's an important aspect to actually realize, that this altogether allows for the passage of solute, a large amount of solute-rich fluid. And I'll call it filtrate. Blood supply to the nephron. Remember, this is a microscopic unit. This is not anything that you would actually be picking out in doing your renal eval. Afferent arteriole brings blood to the nephron and has a larger diameter. Efferent arteriole, smaller diameter, takes blood away from the nephron. And because of this, we have the creation of higher pressures, hydrostatic pressures, inside the glomerulus. So afferent arteriole to glomerulus to efferent arteriole. Then we will give rise to what are called peritubular capillaries. So the efferent arteriole isn't done because it's got arterial blood in it. So that means that it's got still got good nutrient-rich blood in it. And we're going to learn that it's going to we're going to put all the good stuff back into it because it's called peritubular, because it's surrounding the tubule, more specifically the PCT and the DCT. Proximal convoluted tubule and distal convoluted tubule. And so it's going to reabsorb all the lovely solutes that got put into the filtrate and put it back into the bloodstream, and with that also water. There is another capillary, and on the other side of that capillary bed will then be a venule. That's a normal capillary bed. Vasa recta is another capillary bed that's normal, will give rise to a venule when we actually get through that. And it's going to be paralleling the loop of Henle, so a lot of the vasa recta will be actually in the pyramid. And it's important because it's going to help maintain concentration gradients of the pyramid. Juxtaglomerular apparatus or complex, that is indeed where the distal convoluted tubule will come into contact with the afferent arteriole right next to the renal corpuscle. And there's going to be two different appearance to the cells of the afferent arteriole and also of the distal convoluted tubule. The afferent arteriole will have within its wall these cells that are called granular cells. These granular cells are mechanoreceptors. And when properly stimulated, release granules of renin. And that's a very potent vasoconstrictor. The macula densa cells are going to be part of the distal convoluted tubule, and the only way I can try to give you a clue to remember macula densa, which one is it associated with? DCT, distal convoluted tubule, and these guys are chemoreceptors. They detect sodium chloride changes. Delta is changes. Too much sodium chloride, too little sodium chloride. And that's going to modify how much blood flow and therefore filtrate will make. But that's for more of the story. Collecting tubules receive filtrate from many nephrons and empty. Let's go ahead and the collecting tubule is at the end of the nephron, so end of nephron, and empties into collecting duct. And remember, the collecting duct is the one that's going straight on down to the papilla of the pyramid. We're going to get into some physiology now. That was mostly anatomy that we went through. We are going to talk about how we make glomerular filtration. So we're squeezing all of the filtrate and fluid out of your bloodstream, which means your blood ought to just gel up. But it doesn't, because we have then tubular reabsorption. So we take what your body needs and we put it back into the bloodstream. So you don't turn into a jelly. And things you don't want, that you want to get rid of, you will secrete into the filtrate. And so tubular secretion is also a process that is part of what happens with our nephrons and the collecting duct. Interestingly, glomerular filtration is passive and a non-selective process. Passive means no ATPs required, and non-selective means everything but the kitchen sink is going to go into the filtrate. So what that non-selective process means, everything goes in except our proteins and cells of the blood, red blood cells, white blood cells, and platelets. Proteins, these are typically the larger type proteins, such as albumin. Whenever they test urine for protein, they're typically testing it for albumin. And that shouldn't be in your urine. That's net filtration pressure, that's just a shorthand for filtration, is going to be this formula. And you need to memorize this because this is lovely material to put on any test. GHP, G stands for glomerular. HP is hydrostatic pressure. We're actually going back into capillary dynamics right now. I know you guys had so much fun with that. But it's simpler. It's actually simpler capillary dynamics because we're going from arterial to arterial instead of arterial to venule. So glomerular hydrostatic pressure minus glomerular osmotic pressure. OP is the same, osmotic pressure, and G for glomerular. Because remember, hydrostatic pressure is what I call pushing pressure, and osmotic pressure is what I call pulling pressure, related to the proteins that are inside the capillaries as well as the cells and stuff along that line. Plus what I call back pressure. So we're going to subtract back pressure from this net filtration pressure, and C is for capsular hydrostatic pressure. So as you fill the cup with filtrate, that's called back pressure. And if you get the cup completely full, that back pressure will not allow you to put anything else into it. You guys follow that? So our net filtration pressure is glomerular hydrostatic pressure, which happens to be 60 millimeters of mercury. Our glomerular osmotic pressure, here it says 32. We're going to change this and I'm doing this with you guys live instead of pre-lecture because it's in our artwork here. We're going to make it easy, and we're going to make it 30. And capsular hydrostatic pressure, or back pressure, we're going to make that 20. In the old exams, the glomerular hydrostatic pressure was 55. We're actually just kind of updating some of our numbers a little bit, but I like to round them up to an easier to remember. So 60 minus 30 plus 20 would be 60 minus 50. So our net filtration pressure is 10 millimeters of mercury. And that's all we need to make 180 liters of filtrate a day. So if I, for some reason, had a stone in my tube somewhere along the way, what would happen to my capsular hydrostatic pressure? It would go up, correct? It would cause this cup to fill up to its full maximum. And it could only go up to 30, correct? Because that's what we have here is 20. And even though it's 60 on one end, remember, hydrostatic pressure and osmotic pressure and then back pressure. If it goes up to, if our capsular hydrostatic pressure goes up to 30, what does that mean for our net filtration pressure? 0. It means I can't make any more filtrate. And so that's what stones do to a system. Stones obstruct the outflow, correct? So renal stones or kidney stones obstruct the outflow in many areas, and they all start as a granule that starts in the tubular system, and then they flow down the tubular system and settle into the various collecting structures of the kidney, minor calyx, major calyx, pelvis, ureter, bladder, whatever. And so that's an important aspect. Stones, if you're a stone former, that means that if that stone doesn't move soon, what will it do to the nephron, do you suppose? It'll destroy it because you can't get filtrate formation, you can't get the nephron to do its job, and so what's this efferent arteriole doing anymore? And you'll end up actually offing some of these nephrons. So stones are not good for kidneys. So here we'll just change that to 30, change that to 20, make our notes a little bit simpler. Here in our notes, glomerular filtration rate is abbreviated GFR, 180 liters a day, and it is basically all due to that 10 millimeters of mercury net filtration pressure. How do we regulate how much filtrate you make in a day? There's two main methods. One is autoregulation, which means the kidneys have the ability to actually regulate their own blood flow. One of the things too, let's talk about high blood pressure. With high blood pressure, what what number goes up with high blood pressure? Of those three numbers. Yeah, glomerular hydrostatic pressure goes up with high blood pressure, correct? And so that means that if this number goes up, then this number will go up too, correct? And ultimately you'll make more filtrate, but that's not what most people do over time. And that's because we actually have autoregulation. Autoregulation is due to the afferent arteriole. It can adjust its lumen diameter to maintain a constant blood flow and pressure. So what this is called a myogenic, myo is muscle, response. So when the muscle gets distended with high blood pressure, the muscle in the arterial walls, remember it's all smooth muscle, it will actually respond by actually causing some vasoconstriction. So myogenic response, afferent arteriole senses changes in lumen diameter, which is the same thing as blood vessel diameter, correct? The lumen gets bigger, the blood vessel is getting bigger. Senses changes in lumen diameter, and the only reason that the lumen diameter will change is because we're getting high blood pressure, more blood flow, or low blood pressure, less blood flow, and will constrict, which is vasoconstriction, or relax, vasodilation, accordingly, to maintain a constant blood flow and pressure flow. Yeah. Starting to spell like Trump. That's one way we can regulate the amount of filtrate that we do, autoregulation, myogenic response. The other way we can regulate the amount of filtration formed is via our sympathetic nervous system. Because that's what, remember, our sympathetic nervous system is what controls blood vessel diameter. When you send out more impulses, that causes vasoconstriction. When you send out less impulses, that causes less vasoconstriction. And so we will constrict our afferent arteriole with sympathetic stimulation. And when we do that, less filtrate will be formed because this is a narrower diameter, and it doesn't, and the other blood vessel, the efferent arteriole, also is a narrow diameter, so our hydrostatic pressure inside that will actually drop, and you'll make, therefore, less filtrate. This diverts, again, blood flow away from the kidneys. Also, the sympathetic nervous system can stimulate the juxtaglomerular cells of the afferent arteriole, remember those guys, to release the renin granules and increase systemic blood pressure. So sympathetic stimulation decreases blood flow to the kidney and will release renin from the juxtaglomerular cells of the juxtaglomerular apparatus, and those guys are also in the afferent arteriole. Tubular reabsorption. You guys still following me? Okay? Yeah? Okay, so we created this large volume of filtrate, and now we need to reabsorb it back into our bloodstream, or else we're just going to dry up and blow away. So that's what reabsorption is all about. We're going to reabsorb it back into our bloodstream, and solutes will pass through the tubule cell into a peritubular capillary. And so this could be macula densa, capillary bed, or the other one that I'm brain farting on. Not peritubular capillaries. That's why I'm brain farting, because it's so easy. Peritubular capillaries. That's fine. Peritubular capillaries or macula densa. No, what am I doing? Not macula densa. Sorry, people. Yep, no. Vasa recta. There I'm going. You know when you're wrong and you can't remember the right way. Okay, sorry about that. The reabsorption, there's a lot of active reabsorption, which means this is an energy requiring process. And these are the things that require active reabsorption: glucose, amino acids, vitamins, and most of our ions are actually going to be reabsorbed, and the bulk of the reabsorption is going to be in the proximal convoluted tubule. Most of this reabsorption process involves co-transport with sodium ions. So they're coupling diffusion gradients for sodium to also help provide some of the energy for this reabsorption of these various solutes. So they use sodium ion gradient to help provide energy to move these various solutes into the cells of the tubule system. This is going to require carrier proteins. Those are transmembrane proteins with a channel in the center for that. And this is to get solutes into the cell, as well as get solutes out of the cell on the basilar side, and therefore then into the bloodstream. What's interesting about these carrier proteins throughout the tubular system, we have what's called a transport maximum of them. And the transport maximum is going to be dependent upon the number of carrier proteins available, and also the size of the pore through the carrier protein. Like our door here, it's easier to get a lot of people out of our double doors than it is to get people through just a single door. And the more doors you have, the more people you can get out, correct? More double doors, for example. And so that's what this is referring to. This is referring to you actually have more portals in an area to move solutes out into the cells themselves. So let's make a glomerulus, which is just a squiggly of capillaries. Let's make an afferent arteriole. Let's make an efferent arteriole. And then let's make our capsule over this, a little cup. And this is going to be our PCT, but we stretched it out because it's easier for drawing purposes. And let's put in some glucose carrier proteins. So we have a certain number of glucose carrier proteins. And let's make our little glucose red here for purposes of... And glucose is going to actually be part of the filtrate. So it's going to fill up our capsule here. And it's going to come on down the proximal convoluted tubule. And of course, it's going to move glucose out of the lumen into the cell itself. until finally we don't have any more glucose. Now, that's if you have a normal blood glucose level. And people who have diabetes, one of the things that they test their urine for is for glucose. So what that means is that these people have overwhelmed the system because we have only a certain amount of carrier proteins. And now we have a huge number of glucose molecules in the filtrate, and they can get by. We're trying to dilute it, grab them out, but we can't grab enough of them out because there's just so darn much. So that's that transport maximum. That's why you actually have glucose appearing in the urine. Vitamins. Vitamin C is what? Fat soluble or water soluble? Water soluble. So vitamin C is in the filtrate. And when you take a big vitamin C tablet, 500 milligrams of vitamin C, what do you notice? Your pee is suddenly full of vitamin C. And that's the same process. You have so much vitamin C in your system, and you have only so many doors for vitamin C to get taken out of the filtrate that you've overwhelmed your transport maximum, and so therefore now you actually have the vitamin in your urine. So you can only absorb back only the amount that you actually really are supposed to have. So transport maximum is a very important process. Passive reabsorption. Remember, diffusion and osmosis always go down their concentration gradients. And osmosis is something that we always talk about with water, correct? And so water always follows solutes. So as we take the solutes out, water is actually taken out too. But it's taken out via this passive process of diffusion, which is osmosis itself. We don't actually actively transport water. We remember water channels or pores are called aqua porins. Excuse me. A-Q-U-A. Porins. So it really is how much water can flow. It's going to be dependent upon how many aquaporins we have. Just like transport maximum, we have only certain numbers of water doors. Areas that are responsible for the reabsorption of solutes and therefore water. Proximal convoluted tubule is the star of the show, absorbs almost all of our substances, 75 to 80% of our solutes, and therefore water is absorbed. In the nephron loop, we're going to see that it's going to be more important for water movement. But I think I don't want to say mostly control of water, because the PCT is going to be the biggest controller of water, but we're mostly interested in water movement. And the water movement is out of the filtrate back into your system. DCT, or distal convoluted tubule, we will be able to actually take up even more than normal the amount of sodium when aldosterone is present. So reabsorb sodium. So sodium's in the filtrate, we reabsorb it, we put it back into our bloodstream, and whenever we put sodium back into our bloodstream, which is an ion or a solute, we will also get some water following that. The last thing that will happen with our nephron and collecting duct is tubular secretion. So the cells of the tubule are actually able to clear our blood plasma of unwanted substances that are still in the bloodstream. So we didn't get enough of this stuff maybe in the filtrate, and we have too much still in the bloodstream, and so we want to actually pump it into the filtrate, which will eventually become urine, and we can do this to protons. We can do this to potassium. We can do this to ammonium. We can do this to urea. And we can do this to creatinine. There's others, but that's the ones that I want you to know. A lot of people call this tubular reabsorption in reverse. I like secretion is excretion. Excrete or ultimately make urine or urination. Once we get the material into the cell of the tubule system, it will then pass it into the interstitial space, which I don't believe is an organ, but maybe you've heard in the news now they talk about the interstitium being an organ. I feel more like it's tissue related. But we get it into the interstitium, and then from there we get it back into the bloodstream. And it's easy to get it back into the bloodstream because we've actually sucked out a lot of water out of that bloodstream, and so it's kind of a little hypertonic. So it's easy to get stuff right back into it. Yes?
So when I was going through an Old Testament, it said if it became impermeable, so nothing can get through, would that happen then? If the passive would start happening, it would just by osmosis start to go back into what would happen?
If you are impermeable, that means that nothing gets through, so everything stays in the filtrate. If you're impermeable, because we're talking about moving stuff from with reabsorption, we're talking about moving stuff from the filtrate and putting it back into the bloodstream.
Okay,
so if you're impermeable, that means nothing can leave. And so that means 180 liters out of your body.
okay.
Yeah. So that system is not the way it is. But we'll talk about now how we actually change the concentration of our urine. Because if we make our urine more concentrated, that means we're actually producing typically less volume when it's more concentrated. Let's take a look at this. Because we don't want to be dilute. You know, we don't want to pee out all of our good stuff. We want to bring that all back in. And so let's take a look at how we do this. So creating and therefore making a concentrated urine. Blood and filtrate in the upper part of the tubule is at the same osmolarity, 300 milliosmoles. So our blood is 300 milliosmoles, and our first formation of filtrate is 300 milliosmoles. So that's talking about in that corpuscle. Or capsule, into that capsule. When we take a look at, does anybody here eat kidneys? It's an organ meat that not many people eat these days. And in order to eat kidneys, you have to soak them over 24 hours in milk because they are rancid. They're really, they're not why people eat them, I guess, you know, in olden times you ate what was around, yes? But right now we don't have to eat kidneys because we have everything else that we would prefer to eat around us over kidneys. But you had to soak them. And the reason you soak them is that you had to pull out a lot of concentrated material that's actually in the medullary area, and more specifically the medullary pyramids. The osmolarity of our blood and our filtrates 300 milliosmoles at the beginning of things. So take a look here at what the concentration gradient is in the cortex, 300 milliosmoles. Now take a look at this concentration gradient as we go down a pyramid to the papilla of the pyramid. We go to 400 to 600 to 900 to 1200 milliosmoles. That's like going into a salt mine. I mean, it's the saltiest place that you could actually ever want to be. And so that's the one thing about the pyramids of our kidneys. They're highly concentrated salty zones. If you're salty, where does water want to go? Into the salty area to dilute it. So this is a very important thing here to realize. The other thing too is, let's take a look at our nephron loop. The descending limb of our nephron loop is now, here's that impermeable to salts, but freely permeable to water. Our ascending limb is impermeable to water and is actively pumping sodium chloride out. Our collecting tubules are going to be permeable to urea, which means urea is going to leak out of them, and this should be ducts. And remember, our collecting ducts are actually in the pyramids. And is helping to maintain some of that concentration gradient. The vasa recta, which are associated with the loop of the nephron loop, are permeable to salt and water, and they will pick up excess. And return this to our systemic circulation. And that's what we're actually looking at now, our countercurrent mechanism diagram here. We'll go over it quickly, and then we'll I'll review it again in the recording. Okay. Remember, the glomerulus is our blood, and the milliosmoles are 300. The first filtrate is formed is 300 milliosmoles. So we pull off all this solute and enough water to actually keep that osmolarity the same. Take a look at the PCT, and remember, we are absorbing all these solutes, vitamins, amino acids, minerals, ions, glucose. We're pulling a whole bunch of that out of the filtrate. And when we pull all that out of the filtrate, we are also going to have osmosis occur, and water is going to leave. And if you'll notice, our osmolarity of the filtrate, as it's getting less and less in volume, remains the same. So what we take out, water follows with it. So as we then go down into our nephron loop, into a pyramid now, because when we're here, we're actually in a pyramid. This is cortex. This is medulla, or pyramid. When we go down, our descending limb is freely permeable to only water. So we have this 300 milliosmoles, which means we have solute and water in here. And we get down into this area where the surrounding tissues osmolarity is 400. So it's freely permeable to water, and water always wants to go where it's a higher osmolarity, correct? That means there's more solutes there. So suddenly the filtrate is more concentrated as we start to go down. As we get down deeper, our filtrate concentrates even more because more water is leaving. So volume goes down, correct? As we get down deeper, we get to 900. As we get down to the apex or the tip of that area, the osmolarity of that area is 1,200 milliosmoles, and we can make our urine 1,200 milliosmoles at that time. Well, what's really kind of weird, now on the ascending limb, we are no longer permeable to water, and we're actively pumping out solute of sodium chloride. So if we are pumping out solute, that means we're losing solute, which means we're getting more dilute, correct? So suddenly we're 700, we're 400. We're able to pump out so much solute that we become 200 milliosmoles, and finally we're at 100 milliosmoles. That's a pretty dilute urine. And now we're in the DCT, and we are impermeable. Makes it look like we're impermeable, or we're pulling out same amount of solute as we were before. This is the area where aldosterone works if aldosterone is present. Take a look at the collecting duct, 100, 100, 100, 100. That means if we stay at that osmolarity, there is no movement of water out, correct? If we stay at that osmolarity. But this is where the collecting duct is where ADH works. The DCT is where aldosterone works. And that's how we actually will affect the concentration of the urine. Because one of the things about ADH, anti-diuretic hormone, that means make less pee, is what it means to me. Okay? And the way you make less pee is anti-diuretic hormone causes insertion of aquaporins into the luminal side of the collecting duct. And if we put in little water gates all down here, what does that mean to the concentration of our urine? It's going to suddenly become that. The more water gates we put in, the more anti-diuretic hormone we release, the more water gates or aquaporins we put in, the more water leaves the filtrate and goes to the surrounding tissue to actually dilute things on down. Because that's what it is via osmosis. So we'll talk about that in my next recording. So we'll finish this and get a little bit into fluid dynamics and pH. Stop there, and thank you guys for letting me finish this portion.